All right, first of all, I'd like to thank Thermo Fisher for inviting me to this workshop, and also all you guys getting up early and listening to my talk. Let me see if I can. Okay. So my name is Larson. I'm the lab director of United Bio Research. We're based in Salt Lake City, Utah, in the United States, and we develop custom genotyping assays. Genetic testing, or the demand for genetic testing, has been increasing rapidly. This is a figure from a USDA publication that they published just recently. And this is shows the number of genotyped animals included in U.S. genomic dairy evaluations. And you see that 10 years ago it went from a very modest amount till more recently there's 1.2 million genotyped animals being used in U.S. dairy cattle. And not only has the demand for genetic testing increased, but also the value of genetic testing, or the value of integrating genomics into your breeding, has also increased. This is a figure from that same study by the USDA looking at net merit and the increase in net merit over time. And net merit is a index for lifetime profitability that uses actual income and expenses of an animal. But you can see that not only is net merit increasing, but the rate is increasing rapidly. And an animal doubles every 5 years. And so from when they started measuring this, it was $19 per year increase. In the last 5 years, it averaged $85 per year increase. So genetic testing has become is a higher demand, and the value of integrating genomics is even greater. And this is largely due to the decreased cost of the technology and of producing sequencing data. This is a figure just from NIH that shows the decreased cost of sequencing. And you can see that it drops dramatically in the years that new technology has come out. And this big drop in sequencing costs largely drives the demand for genomics, and it makes it so that it's more feasible to use genomics. And it's also driven innovation. And so just in cattle, there's been about one publication per day over the last 5 years on publications on how to use genomics in cattle breeding or in new associations in cattle. And at this conference, we've seen a lot of that discovery. So just being at this conference and going home, there's a lot of new data and a lot of new things that we can use and integrate into our breeding programs. But this provides an interesting challenge for us in genomics and that are using genomics in breeding. And that is that the field changes so fast and the technology changes so fast that we have to be able to adapt to the changes and to integrate this new technology and take advantage of it. And I don't think that we've come even close to come even close to the potential that genomics is going to have in breeding. But this is the primary reason why we bought or purchased the Ion S5. And for my talk, I'm going to go over our experience using the AgriSeq panel and the Ion S5 in developing custom genotyping assays. And the equipment is really, it provides a cost-effective way to get genotype information, but it's also very flexible and scalable. So the designing panels on this is very easy to do, and it's a very straightforward process. And it amazes me how easy the technology is getting. And if you want to design a new panel, it's getting to where it's as easy as just selecting the variants you want and clicking Add to Cart and selecting it, and you have the reagents to run the new panel. So it makes it very easy to get new variants added to your panel and to design new panels. It's also very flexible. So with the Ion S5, you can sequence multiple panels on the same run. It's the same reagents that you use. You can run them on the same chips. And it actually, I think the strongest the strongest attribute, sorry, my mouth is just really dry, but I'll get some water. Sorry about that. So one of the strongest attributes, I think, of the Ion S5 system is the scalability and the throughput capabilities of the machine. So if you're involved in sequencing or in a lab at all, you know that in order to get low-cost sequencing, you have to have high throughput. And you can't run a few dozen samples on the machine and get the cost that you need. And the other thing that you know is when you're designing a panel, you have to have high throughput for the panel to justify the cost of designing the panel. But this equipment actually lowers that barrier quite a bit. So, so for example, if I have 3 panels that I'm running, let's say panel A, panel B, and panel C, and I have 250 samples of panel A that I'm going to run and panel B that I'm going to run, and only 10 samples of panel C, I can actually run 10 samples of panel C, but get the cost benefits of running 500 samples. And this really makes so that we can offer a better service also to our customers, is because we can design custom panels without having the throughput needs to get the cost low. So we created some GBS panels for cattle and for beef and dairy cattle. And currently we can test for over 65 genetic diseases that's found in beef and dairy cattle. We also include variants associated with about 70 traits. Our panels include the parentage test, and we use the recommended 200 ISAG variants. And we also can do gender identification and Y chromosome detection on our panels. So when we meet with a client, the question that we ask is, what is the most cost-effective way to get the genetic data to you that you need for your goal, or your specific goal, or your breeding program? And we meet with clients that really want everything, or a comprehensive panel, as much as we can test for. But we also meet with clients that they really only need just a few variants or a few traits that they need to accomplish their goal and get what they want in their breeding program. And with this technology, then we're able to customize a panel and get it as cheap as we can, specific to the needs of the client. And so that's I think the power of this technology is that it really makes it easy for us to customize things and to generate new panels really quickly, to add new variants as things become available. The other thing I want to talk about on our panel is the different types of mutations that we're able to add. So traditionally, GBS is used to genotype small variants. So the chemistry that we're using, we sequence with 200 base pair reads. And the question that we want to ask is, could we actually detect other types of mutations that are larger than the read length? And also gender identification was an interest for us. So with gender identification, why is that important? So if everyone looks at this, what am I? Am I male or female? Most of us can just look at this and see that it's male. And if we can't see that, we're going to have a really hard time breeding. But the reason why this is important is it adds another layer of quality control. So when we get a sample in that's supposed to be male, or we get a sample in that's supposed to be female, we can actually verify that the gender is correct when we get the sample. And Y chromosome variants are also important in other ways. With Y chromosome variants are used in breed identification, and also potentially could be used to detect free Martinism. This is a condition where Y chromosome DNA is found in female twins' blood samples. But this is just an example of gender identification and why it's important. So we was working with a ranch, and we received 193 samples. And the samples were labeled whether they were male or female. Most of these were female, but we genotyped them and found that 98% of them did match the gender that the farmer had told us, but 4 samples, or 2% of the samples, didn't match the gender. And so that can be from either 2 things. One is that we didn't genotype it correctly, and one is that the sample was mixed up when it came to us. And we did verify these samples, re-isolated the DNA, and genotyped them. We got the same results. But what we found was that the lab was mixing up the samples. And so in this case, for example, we had 2 samples that came in with tag number 25, but we were missing tag number 52. So we suspected that it was probably one was 52. And these 2 samples were genetically different, and this was one of the mismatches. The gender was off on that. We also got multiple samples that came in from the same tag number, and we were only expecting 1 sample. And so this was clear evidence that there was actually a sample mix-up or mislabeling and some contamination that was happening. And this was important for us because we followed up with the rancher that collected these and found that they didn't have very good record-keeping protocols, and they weren't collecting the sample very well. And so it helped us on helping to train them on knowing how to collect the samples and being really careful. And I think the big thing is that they didn't understand how important it was not to contaminate samples. And anyone that's been working with cattle, I don't know if anyone else has seen this, but hair samples, it seems like there's more contamination when people collect hair samples. I think it's just easier for them to mix together. But that's one note for anyone that does hair samples is to just be really careful. You can do it without getting contamination, but you just have to be a lot more careful with them. So the other thing that we were interested in is, could we actually put large mutations on our GBS panel? And probably the most popular mutations in cattle that meet these requirements are the polled mutations. And the first mutation is the Celtic polled mutation, and this is a 212 base pair duplication on chromosome 1. And this 430 base pair region in the mutated allele is too big to sequence across. And so we actually worked with Thermo Fisher to design some primers to amplify either the mutated or the wild type allele. So we designed a primer set that would amplify just the wild type allele, or the normal copy, and then another primer set that would amplify only the mutation, or would only amplify if the mutation was present. And so instead of looking for specific variants, we're actually looking for the presence or absence of specific amplicons on our panel. And we found that this actually worked out pretty well. We wrote a script to just count the number of amplicons that we saw in each sample. And in this sample, most of these are actually wild type, but sample 11 and 12, you can see we detected amplicon B, so these are carriers of the mutation. And this correlated with the phenotype, and of course we verified all this with PCR. The second mutation is the Frisian polled mutation, and this is an 80,000 base pair duplication. Of course, this is way too large to sequence across on a GBS panel. For this one, and then we selected variants across the duplication with the idea that we can detect copy number variation in this. And so we selected about 20 SNPs across this duplication, and copy number detection from sequencing data is getting more fairly common. And I notice on Thermo Fisher's website now, when I go in and design a panel, they actually have recommendations on how to detect copy number variation with your GBS panels. So this is just a few examples, or a few of the results that we had. So you can see that the relative coverage actually goes up with wild-type animals versus animals that are homozygous or heterozygous for the mutation. So you can start to get copy number variation from the sequencing data. Now for us then, the detecting copy number variation is actually more challenging than the other variants. And a lot of that is with the analysis of the data. So with other variants or mutations, when you add to your panel, you just order the primers and you can detect the variant. But with copy number variation, you can't just detect it. You have to have cases and controls so you can get the relative expression between what's normal and what's not, because you get variation in coverage just by PCR bias, and different variants or applicons will actually amplify better than others. So it did end up being a little bit more challenging to add copy number variants to our panel, but we were able to do it. You do have to have good, high-quality sequencing data to do this. You have to have cases and controls, or animals with the mutation, without the mutation, in order to add it. But it does mean that it is possible to actually detect large duplications and other mutations that you wouldn't otherwise be able to add to your GBS panel. So just as a conclusion, so the use of genetic data or genetic testing is increasing, and the value of genetic testing is increasing. And with the rate of discovery, then it means that us that were involved in it, we have to be able to adapt to the environment, or to adapt to the data. And the Ion S5 really provides the flexibility to adapt to these changes. And we can quickly add variants to our panels as needed. Also, we've been able to detect more than just point mutations or small insertions and deletions to our panel. And also, we're working on adding additional mutations to our panel. So free Martinism is something that we're interested in seeing if we could detect using GBS, and also some more large duplications and other mutations that are of interest. With that, I'd just like to thank Thermo Fisher Scientific for allowing me to speak at their workshop, and of course all the beef ranches and dairies that we have worked with. And then I'll just leave it open for questions. And I left my email address if anyone would like to contact me.